Good afternoon. Thank you, Sarah, and thanks to the meeting organizers for having Adaptimmune present the company overview today. We are also a publicly traded company, so our disclaimer slide. Adaptimmune is really trying to lead the way in engineered TCR T-cell therapy. We are based in Oxford. Our research facility is there. We are in Philadelphia, currently building a manufacturing facility in addition to our clinical staff. We have really spent almost 2 decades identifying and validating tumor-specific targets for this platform. And it's really an amazing opportunity because I think we've brought so many targets to bear, which you'll see in subsequent slides. So our SPEAR T-cell platform, SPEAR stands for Specific Peptide Enhanced Affinity Receptor, and you'll see what those targets are. What we've demonstrated is initial solid tumor efficacy with our first target, that's NY-ESO, a cancer testis antigen. And we have 3 other wholly owned proprietary assets, 2 of which are also cancer testis antigens. One is an alpha-fetoprotein target. And the goal for 2018, since these trials are in clinical stage in phase 1-2, is to demonstrate certainly initial safety, but hopefully efficacy in these trials. So why is TCR different than CAR-T? Just to illustrate that really with TCR, we're able to get an addressable population of intracellular peptides. And I think it's an important distinguisher compared to CAR-Ts, who are right now anyway limited to more extracellular targets such as CD19 or BCMA, which make great targets, right, for our liquid tumor populations. But I think that certainly having access to many more intracellular peptides allows us to demonstrate the solid tumor activity that we are seeing in the clinic. And just so everyone's aware, the peptides are basically degraded internally, right, and presented on the cell surface through the HLA complex. So we are HLA dependent in our therapy selection of patients. So I think we've seen this in different iterations today, but what does this mean for the patient with TCR therapy? So if you walk from the top left part of the circle here, we first must identify the appropriate patient. So this is truly personalized medicine in an autologous treatment, right? We're screening for HLA positivity based on certain subtypes, and then we are looking at tumor antigen expression of targets. So we truly are trying to get the patient that could best respond to our therapy. At this point, we would allow a patient to enter the trial through an approved apheresis center, take the apheresis product, and cryopreserve as we ship to a manufacturing facility. We currently work with PCT, but we are also building our own manufacturing facility in Philadelphia in the Navy Yard, which I alluded to earlier. We have a proprietary relationship with Thermo Fisher with the Dynabeads for TCRs anyway, with CD3 and CD28. So we positively select out those T cells, at which point we then introduce the lentiviral vector, which again I think we've heard is a common way of engineering these T cells. Once we expand the T cells to the appropriate dose, we harvest them and freeze the cryopreserved product back to the site, in which time we are able to schedule lymphodepleting chemotherapy, and you'll see what regimens we use in our clinical trials in the upcoming slides. So just in terms of our pipeline, I'll start with the top 3. These are our wholly owned assets. So the first 2 are the cancer testis antigens, MAGE-A10 and MAGE-A4. And the MAGE-A10 population, we have 2 ongoing trials, one dedicated to non-small cell lung cancer. Then we have a 3-tumor trial as well in the clinic looking at urothelial, melanoma, and head and neck cancer. The MAGE-A4 program is a basket trial, and really the utility with that was that MAGE-A4 is expressed to a great extent in a large number of tumor types. So we felt that this was the best approach and hopefully can be a tumor agnostic approach with the regulators. AFP is designed to target the hepatocellular cancer, and that is ongoing as well. The last two on the bottom here are now GSK had a collaboration with us, and they recently exercised their option to take forth these targets, which is great news and I think validation for the platform. And so I'll get into the specifics of the GSK option in another slide. But synovial sarcoma really is our program that's furthest along clinically. We've presented a number of clinical updates with respect to synovial sarcoma, and I'll show you some of that clinical data in the upcoming slides. And they've also nominated PRAME as another target to collaborate. So just in terms of efficacy data and what we're seeing, so this is sort of a summary of the NY-ESO program to date. And in the synovial sarcoma population, this has been an outstanding result. These are heavily pretreated patients. We're talking third, fourth, fifth line of therapy, and we've seen a 60% response rate at the target dose. And a picture speaks 1,000 words. You can see from the PET scan and the image in the middle that truly there is a powerful response when you administer these T cells. And so far, the projected overall survival is at 159 weeks. So additional clinical data here, we've had other trials outside in other indications of synovial sarcoma also showing tremendous response rates in heavily pretreated patients, as well as overall survival rates. We've had melanoma response rates of 55% and a 33% overall survival at 5 years. Again, really talking to the durability of some of these responses. And in terms of multiple myeloma, this was a study conducted in the context of autologous stem cell transplant, and we illustrated a 91% response rate. I think what's an important distinguisher to date, and the N is small, so I'll just caution everyone with that, but the grade and extent of CRS does not seem to be as prevalent when you compare to some of the CAR T therapies. Again, it's a small N and a limited number of tumor types, and our ongoing trials will hopefully continue to qualify that and demonstrate the same degree of side effect profile. We've had 0 neurotoxicity in every clinical trial patient we've treated. So again, I think a unique distinguisher there. So as I mentioned, the GSK exercise of option just occurred about literally a month ago from today, which really, again, I think validates the platform and really provided us with cash runway as well for a small company. So they will provide up to $61 million to us through different milestone payments and including a $39 million option fee. At this point, we're transitioning. We have about 8 months of transition that needs to occur, but GSK will then assume responsibility for all development, manufacturing, and commercialization activities around the NY-ESO program. And the collaboration signed back in 2014 covers up to 5 additional programs. We have already said they nominated the PRAME target outside of NY-ESO, and then they can opt into 2 more. Very importantly, however, to note is that they cannot nominate any active targets that we are currently working on that are wholly owned by Adaptimmune. So just to get in a little more detail of our current study design so folks are aware, if we just look at the MAGE-A10 bucket, again, we've got that triple tumor trial and the non-small cell trial, and these are modified 3x3 study designs, and so we're really looking at enrolling 3 different cohorts based on dose level of cells and expanding based on toxicity. And importantly, we're using modified lymphodepleting regimens, and we really learned a lot in our synovial sarcoma trial exploring a number of cohorts for patients around what lymphodepleting regimen was truly necessary to see the expansion and persistence of the cells, but also to limit, possibly the side effects associated with the lymphodepleting chemotherapy. So you'll see for all of our ongoing studies, it's cytarabine followed by fludarabine for 3 days. So I'd be remiss if I didn't mention our universal cell agreement, really a platform that we're in early stages with, but this is a gene editing technique to remove the HLA class 1 and 2 and the TCR in hopes of really increasing durability with an off-the-shelf approach to these therapies. We also would hope that this would eliminate any graft versus host potential, but certainly an exciting collaboration that we have here in hopes of getting clinical trials sometime in probably 2022. So just to review the year for us, we've really had an exciting year at Adaptimmune. It started with the additional IND filing for MAGE-4, GSK opting for their second target in the PRAME target. We importantly went out and did a raise and had $103 million come in from that raise series that we were able to do so well, I think. And we've presented a number of important clinical milestones for the company at various meetings, and will continue to do so through the rest of the year. And then most importantly, in 2018, really hope to start to demonstrate beyond the safety signals, but see some efficacy in all these additional tumor types. So I hope I've demonstrated today that we have a really active clinical portfolio in our SPEAR T-cell affinity receptors, and that we've demonstrated the initial efficacy and I think unique safety profile compared to the CAR T space. And we look forward to providing additional updates certainly next year at this meeting. So thanks for having me.